let's get back to the gonads and how they work. So we remember they have two jobs. They need to make hormones, estrogen or testosterone, and they need to make gametes, which would be ova or oocytes, your egg cells, or your sperm. So to review from earlier, the hypothalamus in the brain, if it decides we need more hormone, it's going to make GnRH, gonadotropin-releasing hormone, that travels in a paracrine fashion to the pituitary gland. The pituitary gland says, yes, sir, I'll get right on it, sends out LH and FSH into the bloodstream, endocrine secretion. LH and FSH travel to the gonads, where LH is going to stimulate hormone production, and FSH is going to stimulate gamete production. So if we look specifically at the testes, right, they're going to make testosterone by those Leydig cells. It's a negative feedback system. So if the testosterone level gets too low, hypothalamus will make more GnRH, pituitary will make more LH and FSH, and the testes will make more testosterone. If the testosterone level gets too high, then hypothalamus turns off GnRH, pituitary doesn't make LH and FSH, and the testes shut down production. So that's how it works in male-bodied people. Now, when it comes to the ovaries, we have special cells that make estrogen. They're called the granulosa cells. We also have another hormone that's produced called progesterone, and that's made by something entirely different called the corpus luteum. The ovaries, interestingly, also produce testosterone, but most of that is turned into estrogen. And the reality is this whole system is actually quite complicated. So we're going to talk about this when we come to the female reproductive system. We have a whole lecture on it. So things get a little bit more confusing when we get to the ovaries. So the testes are a straightforward example of negative feedback, as we talked about. Ovaries, it's a little bit different. We'll come to that later. Okay, moving on. Now let's talk about gametes and the creation of gametes, which is called gametogenesis, because genesis means creation or birth of the gametes. So the word makes sense. All right. So we have to remind ourselves and think about, for example, what exactly is a gamete, if that's what we're going to look at the creation of. And so I like to think of gametes as kind of half-Z cells, right? Because a sperm needs to come together with an egg to create one whole cell. So each gamete is kind of like a half of a cell in terms of the genetic material that it's going to bring to the situation. So a sex cell is a sperm or an ovum that we use for reproduction. And it's going to contain some, in fact, half of the genetic material, the genes, the chromosomes, the DNA from that particular parent. Because then it's going to combine with the gamete from the other parent to create a new unique individual who has never been seen before, which is really cool. So in order to have this half, right, we call that haploid. It's just a fancy word that means half-Z in terms of the number of chromosomes. So if we remember, we each have 23 chromosomes, like 23 pairs of chromosomes, like 23 pairs of shoes, which means a total of 46 chromosomes. So each of our gametes is just going to have one of each of the 23, like just the left shoe of 23 pairs of shoes, not the whole pair, right? So in order to do this, we're going to need to do something special to make this half-Z special haploid cell. Not only are we going to make these gametes haploid, which is new and unique, but we also want to introduce a little genetic variation into the situation. So we want to mix up the combination of our genes. We want to mix up some of the combinations that we have on different chromosomes so that we can get a lot of variety to choose from. Because the advantage of sexual reproduction is you create all types of various individuals with different combinations of traits, right? That is the beauty of sexual reproduction. So in order to make these special haploid cells that have increased genetic variety in them, we're going to do a special type of cell division called meiosis. Now, if you're having nightmare flashbacks to high school biology and mitosis and meiosis, I'm going to ask you to just hang with me. It's not going to be bad, I promise. Okay, here we go. So when we look at making new cells, cells can only come from other cells. So it's actually a form of cellular replication. We often call it cell division, which is kind of a misnomer, but that's the word that gets used. There are two different ways to do it in the human body. One is to do mitosis, in which case you produce an exact identical copy of the cell you started with. So one cell turns into two identical cells. Or you can do meiosis. And in meiosis, we're going to go through a specialized process to create 4 cells, each of which is haploid, having only half the usual number of chromosomes, and they're all going to be slightly different from each other. So if you remember mitosis, this diagram on the right is a diagram of mitosis with the prophase and the metaphase and the anaphase and the telophase. Right? But the only thing I want you to know is that you're making exact replicas of the parent cell, right? So one parent cell is going to turn into 2 identical, we call them daughter cells, right? We're going to remind ourselves we have 46 chromosomes arranged in 23 pairs. So a 46 chromosome cell is called diploid or 2N, right? So you go from one 46 chromosome cell to 2 46 chromosome daughter cells, both of which are diploid. Oh, yeah, there we go. So meiosis, however, is going to be different. And I'm going to break it down into 4 basic steps because this really gets at the conceptual framework of how meiosis works. Step 1, duplication. This seems completely counterintuitive because the first thing we're going to do is create exact replicas, exact copies of each single chromosome that we had. So on the left side, you can see when we start, before we do duplication, for chromosome 1, we have 2 copies, one that person got from their bio dad, one that person got from their bio mom. Same thing for all the different chromosomes. They're going to undergo duplication, make exact copies of each of those. Now they have double of everything. They're going to hold them together at the center, so now they look like little X's, right? So now instead of 46 chromosomes, we have a total of 92. It seems like we're completely going in the exact wrong direction since we wanted half as many, not twice as many, but just trust me, we will get there. So the first step of meiosis, number 1, is duplication, make exact copies of all of the chromosomes that you have. Then we go into step 2, which is called crossover. So you see here on the top of the diagram on the left, we have a blue chromosome with its exact copy and a red chromosome with its exact copy, right? So each one is holding its copy together so it looks like an X. Those 2 sets of chromosomes now are going to cross over each other, kind of like they're going to embrace because they know they're going to have to say goodbye, and they're going to hug each other. And when they do that, they're going to switch some of their genes with each other, right? They're going to exchange some genes like a token of remembrance, something to remember me by, and then they're going to separate again. So now what was one of the blue chromosomes has a little bit of genetic material from what used to be on a red chromosome. And one of the red chromosomes has a little of the material that used to be on one of the blue ones. So now everybody's a little bit different from everybody else. So that's step 2, crossover, and that's how we increase genetic diversity when we're creating gametes. Step 3, then, we're finally going to separate things. So this is our first division, right? So those chromosomes that duplicated themselves and crossovered themselves are now going to separate from each other, depending on where their ancestry came from. So if they were chromosomes that you got from your bio dad, they're going to go one way, and the chromosomes you got from your bio mom are going to go the other way, and we're going to create 2 separate cells. So now that we've separated those out, now we have 2 cells, each of which has 46 chromosomes. So it seems like we haven't really gotten anywhere yet, but we have increased some of the genetic diversity. But we still have too many chromosomes. So now we're going to do step 4, which is the second division. We're finally going to pull apart from where those X's were held to each other. We're going to pull apart, and so each one of those cells that had 46 chromatids is now going to separate out into 2 cells with 23. So at the end of meiosis, so we did duplication, crossover, first division, and second division. At the end of meiosis, we went from 1 cell with 46 chromosomes. Now we have 4 cells, each of which has 23 chromosomes, with some increased genetic diversity such that no one cell is exactly identical to any other cell. It's pretty cool. So, the differences between meiosis and mitosis. Meiosis is super special. We only do this when we make gametes. All right, and we have these 2 unique events, crossover to increase genetic diversity and mixing up how genes relate to each other, and that halving of the genetic material to create a haploid cell. So we start with a normal diploid 46 chromosome cell, and we end with 4 haploid cells, our gametes. Now, we're going to do this activity in class, so I'm going to skip over it for now. It's too fun to skip entirely, so we'll do it Monday. All right, here's the summary of all the things that you didn't like in biology class, perhaps, or maybe you love them. All right, but the point here is the end goal. So they show both of these, mitosis and meiosis, starting after duplication, right? So you see you start off with 92 chromosomes, right? So they duplicated, you have 92, and then you go through the different phases, right? So crossover, first division, second division. At the end of meiosis, we have 4 gametes, all of which are haploid with 23 chromosomes each, ready to seek out their other half someday in the future. All right, that's it for this lecture. Thank you so much for your patience with myself and my meowing cat. Keep warm, and I'll see you in class.